So, hi everyone. Thanks for the invite to present some of our work, our latest work on computational pathology. And today I'm going to be presenting a platform that we've developed for the community to help grow computational pathology called PHARAOH, or Phenotyping and Regional Analysis of Histology, which is a website that you can access right now. And I'm going to be presenting some of the science behind it, how it works, so you have a better idea of what's under the hood while using it. And in short, it's a weekly supervised active learning platform for computational pathology. So I'm involved in patient care as a neuropathologist, and there's a number of ways that you can diagnose someone so they can get the appropriate therapy at different points in their kind of medical journey. You can do this early on with medical inspection or imaging in the emergency room when the patient first presents, by looking for stigmata of genetic disorders that lead to cancer, or looking at radiology images that are often quite sensitive and specific for specific types of cancers. I call these point of care tests because they're done in a relatively fast turnaround time at the patient's bedside in the emergency room, but often they rely on qualitative pattern recognition exercises by the clinicians. Conversely, on the other extreme, we have very quantitative and objective tests that use discrete values to make diagnostic predictions. These are, of course, much more objective, but can cost quite a bit more, on the order of thousands of dollars, and may have a kind of slower than desired turnaround time, where that information may not be available in the timeframes that we need to take care of patients. And pathology has kind of stood the test of time because it has a nice favorable balance between both speed and accuracy, where we can look at patients' tissue, even just using routine H&E, hematoxin, eosin stain slides, and provide fairly accurate diagnoses that are clinically actionable while we wait for more molecular results. And there are a number of things that we can resolve using H&E that extend far beyond diagnosis. We can subtype tumors often. We can grade and stage them. We can kind of pick what molecular tests are most appropriate for that specific context. Often a lot of the information that we're generating is prognostic and predictive. So we can predict patient outcomes and what therapies they're more likely to respond to. And I think one thing that is often forgotten is just how powerful it is for biological insight. So gaining new insight into how a specific disease may respond to a novel therapy by looking at the cellular types and morphology of the cells. So as we said, it's not perfect. There is some balance where some of the interpretations we make are in fact subjective. So there is an increasing interest in kind of resolving those by using more objective modern tools like computer vision and AI. And the power here is that since we have a large collection of images, we can use modern computer vision tools like deep learning to try to teach computers to make kind of inferences based on patterns of cells and labels in different cancer specimens. And the way to effectively do this is to, because most slide labels are at the slide level, many projects or many approaches will do what's called tiling. They'll break the slide into smaller images, and then they will provide annotations for different regions of a slide. And you can see why this is important. Even at low power, you can see that the level of pink and purple in this H&E slide is highly variable. So not all the regions will be diagnostic, not all the regions will have the tumor. And so these kind of patch level annotations are very powerful in teaching the computer to recognize different patterns, and also help improve the specificity of what patterns may be associated with what disease. So once you annotate or have experts annotate these images, you can then run them through a neural network, and the neural network will painstakingly try to find patterns of pixels that more often statistically align with the kind of labels that that have been given, and through kind of transfer learning and optimization of the neural network. Once these things are trained, you can then present to new slides, and it will use that information it learned during training to make inferences and predictions about what the diagnosis or what the kind of clinical parameter would be predicted to be based on the previous information. And this works extremely well. Here are some slides. These are some of the first slides we've ever analyzed with deep learning, but it's suffice to say that the results were impressive right out of the box, and I think this tool is here to stay. And what you can do now is, instead of having to qualitatively describe the slide, you can have very kind of quantitative percentages about what is on the slide. And at least kind of in our initial studies, this seems to be very, very effective. But it does have some limitations. To develop these classifiers requires a lot of manual annotation. Because you need experts, which are often busy, to label these individual patches. And so there is a kind of a scarcity of sufficient patches that have been labeled, where we can kind of generalize this to all the diagnostic tasks that a pathologist or department would need to do on a day-to-day basis, for this to be a kind of a generalized solution. So what we have is a kind of a growing list of what I call point solutions. So we may have a classifier in a publication that can diagnose, can identify prostate cancer in a specific biopsy. We may have something for lung, but unless we have a classifier for each and every diagnostic task, then it becomes more of a kind of aid rather than a full-blown transformational change in how we diagnose and analyze tissue throughout the whole body. So lots of research is going into trying to solve this problem of data scarcity, and there's a lot of really cool and innovative solutions, particularly around weak supervision. So can we use slide level labels to potentially infer what slides have cancer, what patches have cancer, and not require experts to devote their precious time to annotate individual tiles. And this is a perfect example just a few years ago, where multi-instance learning was used, and what this allows the computer to do is to infer what patches of a large slide have tumor by comparing a large number of slides that were diagnosed as either positive or negative for a specific diagnosis or cancer. So when you can, the results of these are exceptional, with even slide level annotations. This group showed that you can achieve very significant sensitivity and specificity without having to rely on pathologists or experts to label individual tiles. And that has a transformative potential to scale the number of these solutions that we have to reach that critical mass where we can use this every day. One of the limitations that the group mentioned is that because of this subtraction that's required to find these kind of high value patches in this weakly supervised way, the number of slides that you need for the computer to be able to do this is really on the order of tens of thousands. And that creates significant bottlenecks because we may not have tens of thousands of slides for every particular task that we want the computer to learn. Especially for new and emerging patterns, this could be a bottleneck. And also for a discovery approach, you know, we just may not have sufficient data for this to be generalizable across every single task that's required. So we're inspired by these approaches. We're looking to develop kind of complementary ways to kind of fill in some of these gaps that these very sophisticated methods can't reach, at least in the short term. And what we've kind of designed is what we call a weakly supervised active learning model with a important crowdsourcing aspect to the analysis. And we call it PHARAOH, phenotyping and regional analysis of histology. And let's just kind of see how this works. So a pathologist can upload any slide to our PHARAOH platform. This is just a raw image, unlabeled data. The system, as we'll see how this works in a second, will carry out what we call image feature-based clustering, and it will divide the image into smaller patches, and then using image features that it extracts from each one of these patches, it will cluster them to get around 9 clusters of uniform patterns that are found in the slide. And you can see that the coloring here is quite non-random, and it matches some of the histological features that we see on this submitted slide. This information is then relayed back to the pathologist, who provides clustered level labeling of these regions, and that's the weak supervision aspect. So they don't need to label each individual patch, but instead provide patch level annotations, and then those are fed back into the system. So let's see how that works. So imagine the pathologist submits this slide. We provide this kind of we call this a havoc map that basically maps the different histological patterns that the computer can perceive. The pathologist can then analyze these patches and find patterns, and then annotate these patterns and feed them back into the system. So the computer can now learn from these expert interpretations. And this is what the active learning aspect of it is that there's an aspect of weak supervision where we're labeling clusters. And then the active learning part is that the pathologist can continue to update these patches and these annotations until the computer can actually recognize these patterns on future cases. Once this is complete, once this kind of iterative process is complete, where the pathologist is happy that the system is doing what it expects it to do, then those can be shared with the community to kind of scale the level of classifiers available for research use. And this can all happen in the cloud. The pathologist only needs to work with a kind of a comma delimited file or kind of an Excel file, and really just update the labels directly into Excel file and share that Excel file back to us. To help them, we provide a probability of what the system thinks the diagnosis is and how confident it is. So it may not need to continue to provide labels for things that have very high probabilities. And you can see that, for example, this green cluster has a very high probability that the computer believes that this is a clear cell renal cell carcinoma, and by leaving it or updating it, if you want to change the diagnosis, you are kind of simultaneously labeling 1,090 tiles, and we think that is quite an impressive throughput. We also provide, as we'll talk shortly, additional metrics like nuclear density, average surface area, perimeter, and circularity to help the pathologist kind of interpret why the computer felt that specific cluster needed to be demarcated as different. And this is basically the same thing here, just showing how these classifiers evolve. So this is a renal cell case. The first classifier on PHARAOH was a brain tumor classifier, so the pathologist would have no option but to use the brain tumor classifier. And you can see that it got some nonsensical diagnoses here called meningothelial neoplasm. The pathologist can then change those annotations in the Excel file to their appropriate class, and then continue to fine-tune the model. And you can see that after just labeling a few classes, the system now is adapted to correctly classify this slide as having clear cell carcinoma. You'll notice that this fibrocollagenous tissue, this purple area here, is also the cyan region here, and you can see that some classes are in fact transferable, which kind of help reduce the load further for the pathologist. So they things like necrosis, hemorrhage are not things they have to label because they can be borrowed from other classifiers. So we covered how classifiers are produced, but we also want to enhance the consumption of them once they are available. So for every classifier, not only can you have it kind of make these semantic segmentation maps, but it also provides biomarkers both within each clustered area and also within the lesional area of the slide. And basically there's three metrics. You have these deep tissue features that are extracted from each region. You have what we call biologically informed features, so things like cellularity, surface area of nuclei, irregularities, perimeter, circularity that may be helpful in grading tumors. And then we also do we use a popular classifier in the literature called HoverNet that allows you to phenotype the cell types within those regions as well. Are they neoplastic? Is there a strong inflammatory or stromal component? And so you get that information for free by using the developed classifiers in each region. So you don't have to reinvent the wheel for each classifier you develop. And we think that is helps with the generalizability and getting immediate markers by only having to train a few classes. So again, you can try it out today. You just log into PHARAOH, pathologyreports.ai. Once you enter the platform, there's a list of the different classifiers available. You can search for a specific organ site, or you can just browse through them and select the most appropriate one. Once you select one, you get to a page like this. You see kind of a confusion matrix that gives you some context of how it performs across various classes. There's a little description about how many cases were used to make this specific classifier. And then you can either upload your own slide. You can use a sample slide if you just want to try it out. And then we're really excited about the ability to also provide a Google Drive link. A lot of these files are very large, so it could take some time to upload an SVS file that's multiple gigabytes. So you can alternatively just upload it to your own personal Google folder and then provide that link. Of course, these are anonymous slides, and then it will kind of go through the list of SVS files in that folder and generate a report for each one. So all you have to do is click generate. You can go to sleep, you can go exercise, and then once the model is done, it will show up on your dashboard, and then you can download the results and view them at your own leisure. So what you get by running a slide is you get a report, a PHARAOH report. On the first page, you get a kind of a diagnosis, an overall diagnosis or interpretation of what is the most predominant class on the slide. There's a preference to focus on lesional tissue types. So if there's a focus of cancer and a lot of brain tissue, it's going to obviously focus on calling this a cancer. You get a kind of a summary of what classifier was used, what classes are in there, just so all of this is kind of well documented. On page 2, you get what we call a class activation maps that gives you kind of a color-coded map of what different tissue types were found across the specimen, and respective percentages of each particular class based on the classification scheme. You also get approximate surface area. We think this is useful for kind of thinking about molecular studies, and then kind of a high power view of a lesional area. So you can see that this, in fact, the infiltrating component of a tumor. We have this TSNE map that also shows how a particular slide looks like in relation to the training data. You can see that the points out here suggest that this tumor is very different than the tumors in the existing system, just so you have a kind of an ability to see, how these patterns relate to the cases that were trained for the classifier. On the final page, you get this HAVOC map, as we talked about, which is basically a feature, image feature representation of different areas of the slide. And, we find that at least in the majority of cases, different morphologies tend to have a kind of spatial dependencies. And so you get these very beautiful maps telling you what type of tissue you have in more particular areas. So I'm going to show you what you can do with some of the features that are outputted by the system. So deep learning features or deep tissue features. When the slide is analyzed through our system, what we found is that part of the final classification step is done by using these kind of deep features in the kind of penultimate layer of these neural networks. And what we found is that these individual features actually align very nicely with kind of the same features that pathologists may use to make these diagnoses. So we see that this deep learning feature 165 and 66 align nicely with the kind of epithelial component of this metastatic carcinoma, while different features label the luminal components of the same pattern. Other features may label the fibrotic core. So what we think these systems are doing is that they're in fact creating a vocabulary that's very similar to pathologists for the final classification step. And these are very useful. You can either use them as a signature. Here we've taken the clear cell renal cell carcinoma and the papillary papillary renal cell carcinoma data sets from TCGA, and we found that using these signatures, the kind of spatial distribution of these cases are actually quite distinct on this TSNE and dendrogram, highlighting that they have some ability to subtype tumors just using these deep learning features. By kind of pinpointing which features may kind of correlate with kind of ugly looking nuclei, such as this 449, we found that you can also use these features to predict outcomes. Another use that we found is that they are very sensitive to intratumoral heterogeneity. So you have this slide here. This is a glioma slide. This is pure tumor. And you can see that when you run this kind of HAVOC or image feature clustering workflow, you see that the computer has kind of very uniformly demarcated the left and right tissue regions as being somewhat different from one another. And these are in fact are different. So if you measure the cellularity in the red and yellow regions of this kind of tumor specimen, you see that the yellow region has a much lower density. And when you stain for kind of proliferative markers, you see that the kind of hotspots of proliferation match the kind of red area almost in a one-to-one correspondence. So these maps also provide some very interesting kind of biological insights into where biology may be heterogeneous within a kind of uniform tumor specimen. We've also microdissected these areas, and what we find is that the molecular signatures of these areas are quite different. So in the red area, as expected by the Ki-67, we get these kind of MYC pro-proliferative signatures, a lot of kind of undifferentiated stem cell signatures, whereas in the more kind of loose and less cellular areas, we see different signatures, largely related to hypoxia, infiltration and invasion, ECM protein deposition. So we think that that's really interesting, and that could explain why current therapies don't work, is because when you analyze bulk specimens, you can see that the bulk profiles kind of dampen the regional biology that you would get by analyzing these regions separately or using spatial transcriptomics or proteomic approaches, as we show here. So we think that these outputs can also help inform modern molecular studies. In addition to the deep learning features, we also provide kind of biology-informed features, so or handcrafted features, whatever you want to call them. And as you'll see, these have use cases of their own. So to test this, we're now exploring clear cell renal cell carcinoma because there is a very useful grading scheme where the prominent nucleoli and size of the nuclei tend to correlate with outcome, as you see here. And so we thought, can we measure different properties of these nuclei and use those to predict outcome without having to have a pathologist kind of subjectively grade these. So here are some examples of grade 1 tumors. Here's some examples of grade 2 tumors, and you can see that the computer can identify the cells and then kind of compute the surface area. And in fact, if you look at grade 2, 3, and 4s, there is a significant increase in the surface area as you go, as you transition from these different grades. At least in the grade 1s and 4s, or grade 1/2, there's not a lot of grade 1s in the Cancer Genome Atlas, but if you can compare the surface area of grade 2s and 4s, they almost mirror the grades as well. So the 50%, if you divide the grade 1, 2s, and 4s at the 50th percentile mark for surface area, they mirror the kind of prognostic resolution of the grades quite nicely. And lastly, in addition to just looking at tumor cells, you can also look for differences in non-neoplastic cells, such as inflammation and stroma. So we developed this workflow where we use HoverNet to profile the different cell types within these individual tiles. So here's a HAVOC map of this lung cancer, and you can see that the cancer is this dark staining area here that's kind of in the red, and then at the perimeter there's this yellow pattern that the computer proposed that kind of infiltrates into the tumor area. These other areas are kind of lung tissue and some kind of larger vessels or luminal structures. And you can see that the yellow area is predominantly these lymphocytes, and the red area is the tumor. And you can see that, in fact, this tumor has a lot of tumor-infiltrating lymphocytes that can be quantified using this kind of workflow where we carry out the HoverNet cell phenotyping workflow within these different colors. So we can do this across the entire TCGA data set and identify which tumors have tumor-infiltrating lymphocytes and which ones do not. And by quantifying and giving a number to that, this is unpublished work, but we have found that the number of lymphocytes using this approach does in fact correlate with favorable outcomes with higher levels of inflammation in cutaneous melanoma. So this is, again, an example of what can be done using this system to help prognosticate patients. So future features, these have not been developed yet, but we've done research on them, so we're quite confident they work. But just looking at H&E is only one component of the diagnostic process. We tend to use that information to order immunohistochemical biomarkers that we then integrate into the diagnosis. And these are very useful for ordering these more advanced molecular studies. And the advantage of this workflow is, again, it's very excellent turnaround, because you're going from quick diagnostic tests as the H&E to more specific. So you can continually refine your diagnosis, but the patient doesn't have to wait until the very end to get a kind of definitive kind of diagnosis and plan for their specific condition. So now we're trying to automate that second step, the biomarker step. So it's very different than the histological approach, where we're looking at specific patterns. A lot of these biomarkers have brown nuclei, which are positive, and blue nuclei, which are negative. And so we need to quantify those ratios because those have important prognostic significance. And that's called instant segmentation. And there's various tools to do that. We've used MaskR CNN, which basically draws different region of interest boxes around objects on the slide, and then we'll classify them individually. And you can see here that we can quantify a positive and negative cells, and then correlate those. When we correlate those with pathologist kind of estimates, this works fairly well at making fairly good estimates about how many positive cells you have in a particular region of interest. So this is very the nice thing about this is very objective. So if you compare it to the kind of a pathologist concordance, the AI pathologist concordance is usually closer than pathologist to pathologist, because usually the AI kind of usually has a fairly compromise between the kind of overcalling of a generous pathologist versus a more conservative pathologist that may underestimate the score. So we think that there is a great potential for this to be integrated. Just some cool things about how we do this. It's important to note that you can only really measure these biomarkers within the lesional area. So you have to actually use the H&E to find the lesional area, and then only focus the quantification to that region. If these slides were perfectly aligned, you could just use the kind of coordinates to do this, but sometimes these slides have horizontal translation or rotations based on the way they are made in the lab, which requires some kind of creative interpretation by the pathologist to align the positivity with the areas of tumor found on the H&E slide. So the way we kind of overcome this, to automate this, is using image registration tools. We use a classical approach using what's called scale invariant feature transform, which uses kind of low-level features to align two images that have fairly similar objects in them together. And you can see that by doing this, we can kind of overcome these horizontal translations and rotations in the tissue to kind of align those regions and provide biomarker estimates only within the tumor areas of the slide. And so kind of a use case for this is in gliomas, where we once we diagnose a glioma, we then need to molecularly classify it based on IDH and ATRX to either figure out if this is an astrocytoma or a glioblastoma or a oligodendroglioma. And so what we can do is we can identify the tumor based on the H&E slide, as you see here. And then when looking at the IHC, we can use SIFT and this kind of image registration to only analyze the immunohistochemical properties in the areas that are positive. And you can see this is fairly effective. It improves the kind of the prediction that the computer provides. So eventually, we want to kind of integrate that with the PHARAOH reports, where you can get a kind of an integrated diagnosis where you know the specific type of tumor you have, and then have the molecular biomarkers being read out in a kind of a very systematic way, and then have kind of the proof of the work as additional pages of these reports. So a pathologist can review them and sign off on them without having to do too much writing. And this is just different types of gliomas that just to kind of prove what this concept would look like. Now we have a complementary platform called Codito. You may be a computational pathologist or computer scientist that wants to also share their creations. So we've created Codito, which is a system where not only we are uploading our computational pathology tools, but we invite others in the community to also upload their tools, so that others can, just demo them very quickly and see how they work and what their use cases is, and perhaps even use them on a kind of repetitive basis without having to do too much coding. It's important to note that this is a pilot project, so we're interested in kind of the safety considerations. Of course, these are research tools; they're not meant for clinical utility. But kind of thinking forward, we do see using conventional social media ways to help filter out the best performing classifiers. So what we've done, at least on Codito, and it's coming to PHARAOH, is that we note how many people have run the algorithm, and we allow people to provide star reviews and also freehand text about their experience using these algorithms, what conditions they work, maybe provide some insights into what scenarios they don't work. And we think that this will help prioritize the best performing algorithms for the community to use. So in summary, I hope you've learned about new tools that we're developing that could be of value for computational pathology. If you're a coding expert and want to implement your own AI tools and just reach more people, I encourage you to upload your algorithms to Codito. This is not computational pathology specific, so anything that you want to share, please feel free to use Codito as a way to amplify your message. And then if you're a pathologist and you want to experiment with developing classifiers and using them for research, please do check out PHARAOH because we think it's a great way to develop a community of experts to kind of scale AI. Please join or tell your friends to join the PHARAOH community. We have a number of people around the world developing algorithms. We'd love to see this grow to a point where we can really find a solution for each and every use case in pathology. So thank you for your attention. I want to thank the PHARAOH Incudito team that helped drive this work, our funding agencies, and of course you for listening and the questions and feedback you may provide. Thank you.